So right now we're going to start with food packaging and irradiation, which is going to be given by Dr. Martin Hoagland. Martin?
All right. Hello. My name is Martin Hoagland. Thank you for participating in this year's FDA Nutrition webinar series. For some of you guys, it's a Super Tuesday. And I'm here to talk to you today about food packaging and irradiation. So if you weren't asleep before, you might be in a half hour. All right. So many of you that are online right now are nutritionists and dietitians. So I hesitate to mention that one of my favorite foods is Kellogg's Fruit Loops. So I ask you this question. I'm going to open up the poll. How many food additives are there in Fruit Loops? Are there A, 0 through 10, B, 11 through 20, C, 21 through 30, D, 31 through 40, or E, over 40? Now, one thing to consider is that there's 6 different colored Fruit Loops, so you might go with 6. Or you might count up the number of ingredients on the ingredient label, which is 34. So that would put it at D. Now, how many of you, when thinking about this question, thought about the food packaging and the potential migrants from the food packaging into the food? So, in Fruit Loops, as well as many other food packagings, you have a plastic bag or plastic container, potential polymers migrating from the food packaging into the food. You might also have resins that are sealing the bag. In certain situations, you might have direct contact between the box and the food material. You might also potentially see ink from that package migrating into the food. If there's coating or an adhesive in the packaging, that also has a potential to migrate to food. So let's go ahead and take a look at the poll results. It looks like the majority of you had selected E as more than 40. So that's good because it considers the potential food additives through food packaging. Okay, so the Office of Food Additive Safety within the Food and Drug Administration evaluates the safety of food additives. We have 3 different divisions, one that considers direct food additives, and that is called the Division of Petition Review. And commonly, these food additives are evaluated at human exposures of 1 part per million or greater, which translates to be 1 milligram per kilogram food and larger. And these are, this is a pre-market review process, and a common example is pictured at the top right of your slide and includes sugar substitutes such as aspartame, saccharin, and salvia. Other examples might include antimicrobials and color and flavor additives. Secondly, within the Office of Food Additive Safety is the division that covers biotechnology and the generally recognized as SAFE program. Lastly, we have the Division of Food Contact Notifications, which considers food packaging and the exposures at 1 part per million and less. So why is food packaging regulated? Food and drugs began to be regulated in 1906 under the Pure Food and Drug Act. And through time, concern grew over food packaging and food additives. Now, a food contact substance is a substance which comes in contact with food, is not directly added to food and does not have a technical effect on the food. Upon submission of a food contact notification by a company, the Office of Food Additive Safety is required to conduct that review once the submission is completed at 120 days. Now, the safety standards for food additives is the same, whether it's through the Division of Petition Review, DFCN, or what's called the TOR, or Threshold of Regulation, which I'll talk to about a little later. Now, again, commonly with the FCN program, exposures are at less than 1 part per million. With antimicrobials, the safety factor is usually 1/5, and thus the FCN program reviews the safety of these at exposures of 0.2 parts per million and less. So the FCN program will consider indirect food additives. And as I stated earlier, these are substances that have contact with food, have no intended technical effect on the food, and include packaging. All right, indirect food additives can be those within packaging, but they can also be used in machinery. And here we have production lines, and indirect food additives could also contain secondary directs, which means substances that are directly added to food but by an indirect method, such as lubricants within machinery or cleaning solutions on boilers, in which even though the boiler cleaning solution is rinsed out, you assume a 1% residue within the boiler as a potential exposure to the food. So food contact substances are part of a food contact hierarchy. So a food contact article is a finished product and can be composed of multiple food contact materials. And food contact materials can be comprised of multiple food contact substances. Therefore, you can see how the pyramid scheme would work, and you could potentially have a very large number of potential indirect food additives in your food. Now, the safety assessment of food contact substances is comprised of 3 different evaluations. The first part is by an exposure estimate, which is conducted by our chemistry team. There's 2 ways to approach this. In order to calculate an exposure level to humans, one could assume 100% migration of the food contact migrant from the substance into the food, which is a conservative method, or you might actually go and conduct what's called migration testing in order to determine the level of the migrants that move from the food contact substance into a food simulant. Chemistry also considers whether this single exposure is significant and whether it will significantly increase the cumulative exposure when compared to all the other notified uses and the uses that are regulated through food. Now, with toxicological testing, our tox review team uses a tiered review approach in which we consider carcinogenicity as a primary endpoint because it's extremely sensitive, and commonly with a food contact substance, exposure is very low over a lifetime. At higher levels of greater than 50 parts per billion, we may consider non-neoplastic endpoints. We also have a team that evaluates, has a potential to evaluate food contact substances based on structure similarity to known carcinogens or potential carcinogens. And we also are able to conduct structure-activity relationship, looking at similarity to known neurotoxicants or teratogens. Lastly, we also have an environmental review team that reviews the safety and the potential impact of the food contact, notified food contact use on the environment. So for the exposure calculation, as I talked to you earlier, chemistry can conduct a worst-case scenario of assuming 100%, or they may conduct migration testing using food simulants. And using modeling, they calculate a migration level and dietary concentration and estimated daily intake exposure for humans using consumption factors and diffusion coefficients and certain assumptions, including 60 kilograms per body weight for a human and 3 kilograms of food and water consumed per day per person. Now, there is online a CEDI and ADI database if you're interested in determining the exposure levels that are calculated by CFSAN and OFAS. So for the general guidance of food safety, food additive safety, we take a tiered approach. You can look at the left and see that there's different levels of testing recommended for different levels of exposure. And on the right side, you can see the various toxicological assays that are of interest that are recommended at the various exposure levels. Okay, so this is a highly simplified cartoon slide of risk assessment in which we're looking at a non-neoplastic endpoint. And these may include weight loss, death if the multiple animals are dying, but usually you see a non-neoplastic effect before that. You might see an organ weight loss. And so on the right side curve, you will see an effect which becomes statistically significant at a certain level. Depending on the number of animals used within the study, you might draw a level of, at 5 to 10% as statistically significant. And this becomes the NOEL, or no observed effect level. And then using this number, we will determine what's called an ADI, or allowable dietary intake. This uses different factors, including a 10-fold inter-individual variability within humans, as well as a 10-fold factor extrapolating from humans to animals. Now, with carcinogenic endpoints, because carcinogen exposure has been shown to correlate in a linear risk from exposure to risk, we are able to calculate a lifetime cancer risk using what's called a unit cancer risk, in which at a certain dose that we see an increase in tumors at the lowest dose, we will use that point of departure and extrapolate it to the point of origin on the graph. Determining the slope gives us a measure of the potency of the potential carcinogen, and then we will use that number and multiply it by the exposure that chemistry has provided to us. And with lifetime cancer risks of less than 1 in a million, we consider this risk to be negligible increase in risk compared to background levels of cancer within the human population. The next slide, this is not a real molecule, but this shows a structure-activity relationship in which there are known chemical moieties that are indicate potential concern for carcinogens, and these are what we look for in food contact substances. Also, we have a threshold of regulation exemption, which is for extremely low levels of exposure, and guidance is available online. There are multiple forms of food contact plastic. If you look at the bottom of your plastic food containers, typically there is a recycle number, including a number, a symbol, and small little numbers which indicate what kind of plastic. For example, there's high density polypropylene ethylene, or terephthalate, which is 2, and polystyrene, which is number 6. So the FDA has also considered recycled plastics, and our thoughts on recycled plastics is available online at this site. We have no safety concerns regarding recycled plastics. Also, regarding bioengineered food containers, there are examples in which food containers are manufactured out of biomaterials, and the FDA does not have a safety concern for these. Keep in mind that commonly, if they are made out of a biomaterial, the exposure through the diet is going to be much more in consideration to that used within a food container. So the Office of Food Additive Safety has a strong group of talented toxicologists. I'm not including myself in that group. But there's a Tox 21 initiative within Europe, and whose end goal is to reduce animal use. And there are a lot of highly sensitive, very potentially powerful techniques used within science that have been developed within industry and academia. And we would like to potentially use these methods in order to help reduce animal use in testing and help speed along the process of safety evaluation. Next, on food irradiation, in the early 1960s, the FDA determined that irradiation of food is safe. However, irradiation of food does not replace good sanitation and hygienic food production processes. If you look at the symbol at the bottom of your slide, that is called a radura, which is a symbol that is used on food packaging to indicate that your food has been irradiated. Restaurants, however, are not required to use this symbol on their menus to indicate whether the food has been irradiated or not. Okay, lastly, microwave cooking. Microwaves is a form of irradiation, including electromagnetic resonance. It heats food by exciting water molecules within your food. And microwaves actually reflect the microwaves that are produced by the metal coating on the inside of your microwave. The FDA does regulate the emission standards within microwaves. One cautionary use for microwaves: when you're heating water, you might use a small wooden stick or a substance to help break the surface of that water. There are examples in which if you superheat the water, upon breaking that surface, it can be very volatile and cause thermal burns. So that's all I had. Are there any questions?
Okay. It looks like we have several questions. Just give me a moment to scroll back to the first question, as I want to make sure that the person, we're going to take these in the order that they came in. There's a lot of discussion here about things that were in the slides that are not questions. Facts, someone said that Fruit Loops spelled F-O-O-T, F-R-O-O-T, for example. Let's see. Oops. Thanks. All right. We're still trying to get the questions. Thank you. Now, this is one, this is a statement about food additive petitions. And I think I think I'm going to move to, it says, do I need a food additive petition for this additive? I think, do you want to address that, Martin, so that, they can go to the website or find out the answer to that? Says,
Now, I'm not sure exactly what the question is. If you contact the Office of Food Additive Safety, we will be more than willing to provide guidance as to which process you might want to submit a notification or petition for. The GRAS process is different than a petition review, as is the food contact notification, which is also different, as is TOR, which I mentioned. So.
And there's another question that, does FDA consider a synthetic effect of the additive from packaging in the risk assessment?
That's a very good question. At this time, we consider risk to be additive. However, I think when you talk about synergy, you're commonly thinking about multiple compounds and the exposure of multiple compounds and the effects that they may have. Right now, scientists, both regulatory and in academia, are struggling to answer those kinds of questions, because you can quickly see that with multiple compounds in mixtures at different concentrations, one would have a plethora of concentrations and mixtures to test. And there was a really good seminar earlier, I guess it was last year, hosted by NIEHS, that showed that a lot of the effects that are considered tend to be more additive. However, in our safety assessment, we only evaluate the safety, the exposure of a single compound. Another interesting highlight that I'd like to point out to you is that we here in the FDA on the food safety side in food packaging, when we consider plastics, commonly run across information not only in the public literature on the compounds of concern or potential concern. We also have in-house data not only from food safety and what's been previously submitted to our agency and center, but to other centers within the FDA, including excipients and drugs and the colors that are used in drugs. Also, medical devices commonly are made of plastics, and so we have information from that. And there's also compounds that occur within cosmetics that we have information on. Granted, it's a different route of exposure, but sometimes there is information that's submitted from cosmetics that we are able to use as in dietary exposure risk assessment and evaluation.
Okay, moving on. We still have time for questions. Since I just want to scroll back to. There were some. I just want to make sure I get some of the earlier questions. What it looks like, this one says, yeah, there we go. What about irradiation of dietary supplement ingredients?
So there's a question on irradiation of dietary supplement ingredients. I also noticed a question about irradiation of spices. Now, spices are commonly irradiated. There's a potential concern for microbial contamination, and irradiation is an effective method in killing those pathogens. I would assume that Similarly, they should be effective in killing microbes within dietary supplements. However, dietary supplements is a subject that could be addressed by our Office of Nutrition Labeling and Dietary Supplements much better than myself. So maybe they would be able to ask or answer that question for you.
We have a yeah, we have a dietary supplements presentation coming up later today. There's another question. We have time maybe for 2 more questions, and I'm hoping I'm getting to the questions as they came in. I don't want to, yeah, there we are. So there's one about, what about migration of BPA into canned foods? And then, of course, another person chimed in and research and outcomes of BPA. And then would a BPA ban review still be held by the end of this month, something like that. So there are 3 questions, and perhaps you can just address that one.
All right, so I knew this question was going to come up. I'm not directly involved in the risk assessment of BPA. However, our current thinking on BPA is available online. So if you reference that, as far as I know, yes, whatever meetings that are planned on BPA are still going to be taking place. If you consult the website, you should be able to find that information. I don't know it off the top of my head. One thing to consider in the safety review of BPA, so the Food Contact Notification Program evaluates dietary exposure. And obviously, bisphenol A has been on the market since the early 60s. And we have a lot of information, toxicological testing on BPA. We also have pharmacokinetic data that shows that when administered to animals through such as intravenous or intraperitoneal administration, there's a certain pharmacokinetic profile. However, with dietary exposure, a substance would go through the gut, be absorbed to the liver, potentially be metabolized, and then excreted out. The data that we have shows that BPA is quickly glucuronidated within the liver and excreted out by the kidneys. However, so that's one part of the FDA. The other part of the FDA includes medical devices. So there's the potential concern for BPA exposure through medical devices such as neonates or premature babies that are born that are hooked up to multiple tubes in which blood or drugs are administered. And so there's a potential for an intravenous BPA exposure to a neonate or a sensitive population. If you go back to our assumptions, the dietary risk assessments are typically based on a 60 kilogram adult human that's consuming 3 kilograms. Obviously, the BPA review for medical devices is different from that used for food. Now, the industry is moving away from the use of BPA, and the FDA is supporting that move.
All right, thank you.